All right, eukaryotes must process mRNA before it leaves the nucleus. This is something that prokaryotes don't have to do. Why don't they? Well, let's think. Does the DNA ever leave the nucleus for prokaryotes? No, because there's no nucleus. But eukaryotes, we protect our DNA inside the nucleus for a reason. Outside in the cytoplasm is dangerous for DNA and RNA. There's a bunch of enzymes that would basically degrade the mRNA. So what our body has done is we basically put protection on this mRNA. The protection is the 5' cap and the poly A tail. The process of putting these on is called RNA processing. Okay? We also do something in RNA processing. We splice it, and these are called the exons and the introns. Parts of the mRNA are not used to actually code for a protein. So here you see DNA. This is the original DNA strand. Then we added the 5' cap and the poly A tail. And again, this protects it from the harmful enzymes in the cytoplasm so that this does not degrade. And then you see these words, exon and intron. Exon is the part of the gene that is, we say, expressed. And it gets to exit the nucleus. Intron, with the I, is the part of the mRNA that gets removed. It's what we would say a non-coding region. Okay, so when it's done, if you look, these introns are removed and it goes out in the cytoplasm with only the exons and the poly A cap or the 5' cap and the poly A tail. Okay, so these two are basically the job of the 5' cap and the poly A tail is to protect the mRNA from the enzymes in the cytoplasm. And that's what this slide is saying. What would happen to the mRNA if we removed its 5' cap and poly A tail? It would get eaten alive, or not really. It would get degraded by the enzymes in the cytoplasm. Exonucleases may digest since, here, this is the answer. Okay. Which of the following does not occur in prokaryote gene expression but does in eukaryote gene expression? Go ahead and pause so you can go through the selections and answer it. The answer is going to be C. In eukaryotic expression, we have a poly tail added to the 3' end and a cap to the 5' end. Okay. All right, so what are these exons and introns? Well, introns are kind of the junk that we don't want. Exons are what is expressed, or the real part of the genes, okay? So this blue in this picture is showing you the exons, and the orange is showing you the non-coding regions which get removed, which are called the introns. So here we see the introns are removed. This is taking you through the process of mRNA getting edited. The introns get removed. Then we only have the exons left, which you see right here. The little green guys are the introns. The exons are the light blue, and the exons come together and we have our finished copy after it's gone through RNA processing. Here's a test question. There are many non-coding stretches of nucleotides present in eukaryotic DNA. Explain why the original transcription unit is so much longer than the unit used to make a protein. That is due to RNA modification, right? And what that means is those non-coding regions called introns are removed. So your pre-mRNA is always going to be longer than the finished copy of mRNA. Okay, This is just showing you this whole idea of mRNA splicing, that we edit out the introns and pull them out, and that makes the mature mRNA, okay? The things that actually splice the exons, that's something to look at. I mean, the introns. There's two things. The snRNPs, they make up, if you look here, these little guys right here, you see them. They're the little swirly guys. They make up these circles with the swirlies in them. These are snRNPs. A bunch of these together make up what we would call a spliceosome. And the spliceosome is able to recognize that intron site and cut it and resew that part of the mRNA together. Okay?